And what we have here, coming up next is Ravi Sheth. So Ravi is actually one of our own bioengineering undergraduates. So he's 20 years old, he's just starting his junior year, and he was already in the science community. He was working here in Jeff Tabor's lab, but he was newly exposed to the maker community. And so he developed a different way to approach this total ecosystem, but had very similar, very successful results. So, Ravi?
Yeah, so as Jordan mentioned, I kind of came to the maker community from being in the science community for a while. And so my work is with building a cell extruder very similar to Steve's work, except we're using kind of a different method. And so I've been working in Jeff Tabor's lab for the past 2 years since my freshman year. So kind of as a background, you know, everyone else has been talking about how they joined this maker community. I think for me, the first exposure to this was in high school. I had an AP chemistry course, and one of the assignments, extra credit assignments, was to build a battery. So I went home, I went through all of my different things. I looked for anodes and cathodes, I found coins, I found different materials to act as the buffer in between them, I looked for containers. And I really had to research, I looked online, and putting it all together, I really found it was amazing that you start with all of these different materials, you do this research, and then you can finally make something. And I made a little battery that turned on an LED for a couple of weeks. And it was just such an enthralling experience to take all of these parts, put it together, and it was something I made. So as Jordan mentioned, I've been kind of coming from the research perspective. So in high school, I did a lot of research in looking at structural biology. So I did a lot of research in looking at how proteins adapt thermally to different temperatures, or how flexibility in proteins allow them to adapt to different temperatures. So this was all supercomputer simulations and molecular dynamics simulations, more at a structural biology level. But for the past 2 years, I've been working in Jeff Tabor's lab, and what we do is we build synthetic genetic circuits to look at how bacteria signal and how we can use that to create new circuits and understand how biology achieves a level of complexity. And so what we really do is we exploit the central dogma of biology. So basically, you have DNA, which is the genetic code, and it's converted to RNA, which is then translated into proteins. And so basically what we can do is we can exploit this much in the way that you use electrical circuits. So when you have electrical circuits, you start off with all of these different parts. You can put them together in different ways, and you have a design process for doing that, and you end up with an electrical circuit, a computer, a smartphone, an iPad. And much like that in synthetic biology, what we're doing is we're taking different genetic modules, putting them together in different ways. We can basically mix and match the way they work together and create different type of biological circuits. So in this example, it's a 4-input AND gate, and that's one of the most complex synthetic biology circuits that's been built to date. And so really it's a top-down engineering process that's being applied to biology. So you have libraries of different parts that people make, and you have a formalized design process to put them together. And so what I'm really interested in is being able to print genetically engineered cells in 3 dimensions. So the idea behind this is that if you can take a genetically engineered E. coli cell that contains one of these synthetic circuits that we're building, and you can place them precisely in 3 dimensions, there's a lot of different applications. So you can build things like complex biological sensors, or even computers by printing different types of bacteria next to each other and looking at their communication. And you can also start to look at a lot of basic science research into how these bacteria interact with each other. So things like biofilms. Even in our bodies, the way our cells are spatially differentiated are really important to the function of our organs and the function of our bodies. And another really interesting application is if you can take bacteria that take really readily available materials. So for example, something like dirt, and then the bacteria can eat that, and you can engineer them to produce some type of valuable resource. And so an example for this would be if you're on Mars, you take a bunch of Martian dirt, you have a bacteria that takes that Martian dirt and converts it into plastic. Then if you can 3D print that bacteria, 3D print that solution of bacteria and dirt in a specific pattern, you can start to build structures. So you can build habitats, you could potentially print food and things like that. So that's kind of the motivation behind building a cell extruder. And so with this, we had a lot of different engineering constraints, but the ultimate solution was the cell extruder And so basically it's really simple. All it is a stepper motor that's coupled to a shaft, and all it does is it drives a 20 milliliter syringe. So it's just a simple syringe extruder. But the really cool thing is that this was all built with 3D printing. The parts are only $10, $10 to $15, and all the other parts are 3D printed together. And then on the flip side, you can attach a syringe tip, a Lunar syringe tip, to a 3D printer chassis. And so this is the setup we have currently. So I have a bunch of board bots that are an open source 3D printer. And you can, all this is it's mounted to the OrdBot, and the OrdBot provides the 3-axis XYZ linear control, and you can control the syringe independently. And the idea is that in the future you could build 4 or 5 of these, mount all of them onto the same printer, and then print 4 to 5 different strains of bacteria. So what we've been doing to quantify and qualify this setup is just doing some basic experiments. So all we've been doing is printing bacteria that express GFP of fluorescent protein. And so this was one of the first things we did. This is the name of one of the grants we have in our lab. But all it is 2D printing of the cell solutions. So it's E. coli suspended in LB. And the print tip is suspended above an agar plate, and you print directly on it. We're also looking at quantifying this on a microscopic level. So this is fluorescent imagery of the line that you saw in the last picture. This is on an agarose pad. And so this is around 1.5 millimeters thick. We're currently looking at making that smaller by using different tip sizes. But if you look at this on a single cell level, it's really amazing how good of definition you have on the edge. So on the left you have single E. coli cells, and on the right you have this very defined edge. And so as far as documenting and deploying, I've already posted this to Thingiverse, and it's gotten a couple thousand views. But the really cool thing is that now this is out in the ecosystem for other people to look at, for other people to understand what we're doing in our lab, and for other people to reuse for things that aren't even related to science or our research. And so what's really interesting to me is that I think in science you have a really linear way of conducting research. So you start off with your background, you have a hypothesis, and you do experiments, you have a conclusion. And it's really incremental knowledge. You're starting off with this big pool of knowledge, and you're making a small result that kind of pushes forward our understanding. And you publish a paper, you get a patent for the work you're doing. In the maker community, you kind of start off with the internet. That's supposed to say the internet. So you start off with the internet, you have this whole jumble of things going on, and you kind of meander through, nothing's really documented well, and you end up with something cool at the end. And maybe you post that on open source, maybe it just sits in your garage and you show it to a couple of your friends. But I really think that both of these can come together in a really cool way. So why should we make in research? Well, so the really interesting thing is that you have this kind of approach that you make things, you create something from scratch, you reuse things. And in science, you kind of, you're just looking to advance knowledge. But if you can build your apparatus to specifically ask your right questions, rather than kind of just incrementally advancing what you're looking at, you can start to look at bigger questions and make bigger findings. I also think it's really interesting as far as outreach and awareness. So if I'm posting this somewhere online where thousands of people can come and look at it for themselves, people can understand what we're doing in our lab, why they should provide government funding for the research we're doing. So it's not like we're just sitting upstairs on the 8th floor kind of doing our own thing. It's open source, it's kind of released to the community. And really this is being contributed to the community so other people can reuse this design for something completely different from printing bacteria. And it's really something that benefits the greater public. So with that, I'd like to take any questions. Thank you.
So I know you said for the 2D you're starting with the broth with the bacteria mixed in. What sort of materials would you look at to make it 3D? Or is that something like for future work?
So that's kind of the next step, and actually right now we're working to design an extruder that's heated. So the material I'm looking at right now to print with is agarose, or either agarose or agar. And so they're both gels, and you can bring them up to melting point, and they'll stay gelled up to around 30 degrees Celsius, 30 or 40 degrees Celsius. And so all you can grow E. coli suspended in those. And all you do is print these kind of agarose or agar solutions and then cool them and then print another layer on top. So that's kind of the first step, was kind of just quantifying and qualifying, even making a simple 2D cell extruder, and the next step is to go to the 3D level. But agarose and agar is the materials we're looking at right now.
Were you able to quantify your kind of volumetric resolution with your extrusion?
Yeah, so I think I haven't had a chance to do that, but that's definitely something that is the next step to say, okay, I'm going to input this machine to turn the shaft this many times, and we can quantify how accurate you are and how repeatable your results are. So that, yeah, that's definitely something that needs to be done moving forward.
What are options for scaling this into like periodic systems or like mammalian systems? Like for cells?
So I mean, I think Jordan Miller's work is a perfect example of that. So I think it's a pretty similar concept, except he's using a completely different growth substrate, and I guess it's a negative. So you're building a mold and then you're printing in there. But I think you obviously have to change the material you're printing with, so your support structure. And that might require a different type of extrusion method, just because like a lot of the materials might be need to be heated or might need to be photopolymerized.
So along those lines, have you been looking at scaling down to get higher precision, or are you looking to scale up to make much bigger structures, or something that can do both?
I think at this point we're really interested in scaling down, just because a lot of the applications we're looking at are we want to have microcolonies communicating with each other. But I also think scaling up is really interesting too, because if you can get these bacteria, engineer these bacteria and produce substrates from really readily available things, then you can imagine having this gigantic 3D printer taking a bunch of corn husks and printing plastic and making a house or something like that. So I think both directions are valid ways to go, but we're interested more in the microscopic scale of things.
All right, thank you, Ravi, again.